evening, everyone. It's wonderful to see so many folks with us this evening. My name is Jerry Bowers. I'm the Director of Development and Public Affairs here at the MDI Biological Lab. And it is my pleasure to welcome you all to our Science Cafe this evening. We are thrilled to have Dr. Elizabeth Marnick with us this evening to talk a bit about vaccine development, particularly how microRNAs are playing a role in really changing that process of vaccine development. So it's a wonderful technology that has been used extensively to really revolutionize biomedical research over the last decade. And so we're very excited to have her with us this evening, as well as have all of you. So I want to just quickly see if I can pull up my notes here, sorry. I want to just share a little bit about Liz's background with you, because she comes to this with a lot of experience in immunology. And so Liz received her PhD from Tufts University. She was the first graduate, I think, Liz, if that's right, from the joint Jackson Laboratory Tufts University PhD program. So that's a wonderful thing for the state of Maine. Liz did her graduate work at the Jackson Laboratory with Dr. Derry Ropinian, and again, studying the immune system there, particularly infectious disease, autoimmune disease, and so she has an extensive background in this area. She then moved on to do her postdoc with Dr. Dustin Updike here at the MDI Biological Laboratory. So Liz is well known to us, and we are thrilled to have her back. She is currently an assistant professor at Hassan University, and I think if you happen to follow her on social media, or maybe you have a student who has participated in one of the many courses that she teaches here at the MDI Biological Laboratory, you will know what a fantastic teacher she is. And so we are really looking forward to this conversation with her this evening. So join me in welcoming Liz. Thank you so much.
Thank you so much for having me. Let me just get my screen going. All right, I assume everyone can see my screen and the full slides, not the presenter mode. You think by now we'd all be good, but still errors happen sometimes. So thank you very much, Jerry, for inviting me. I'm really glad to be here talking about this important topic. A few things I want to mention before I get talking about this topic is I want to just have a disclaimer right up front that I have no financial ties to any of these pharmaceutical companies that have developed these vaccines. The purpose of this talk is to give you information about how these vaccines work, why they're so revolutionary, answer some common questions that I see people asking about these vaccines, so that you can make an informed decision when your turn comes up to hopefully get this vaccine. And of course, everyone's unique medical history is very individual. So while I can give you general information based on what's known in the research, you should, of course, double check any of specific medical information with your personal physician. So the overview of how I'm going to talk about this topic is, first, I want to give everyone a brief overview of how the immune system works in general and how the immune system then responds to a vaccine. Then I'm going to make sure everyone understands the relationship that exists between DNA, mRNA, and proteins, because I think that's important for people to understand how these vaccines work. Then I'm going to talk specifically about mRNA vaccines, and then I'm going to address some of the common concerns that I hear people express about whether or not they want to get this vaccine. And then, of course, there'll be time at the end, if I haven't covered your question, for you to ask your question as well. So let's get right into talking about the immune system. So when you're looking at the immune system, we can break it down into 3 levels of defense. And the first level of defense is our barrier level. So we have our skin and our mucous membranes that are existing to help protect the pathogens. And by pathogen, I mean things like bacteria and viruses that can make us sick. These barrier levels help to protect us from even getting those bacteria or viruses inside of us. But we all know that we still get sick, so those barrier methods aren't 100% effective, and we do still get infected with bacteria or viruses. So then at that point in time, what ends up happening is that we have the innate immune system, and then we have our third line of defense, which is our adaptive immune system. And I'm going to briefly summarize the innate and adaptive immune system. I'm obviously going to leave a lot out because there's a lot of information about the immune system, but I just want to give you enough information so that you can understand how these vaccines work. So when you're looking at the innate immune system, you have a bunch of cells that are within the innate immune system. And these cells are your first line of defense when a bacteria or viruses penetrate that barrier and infect you. And I like to equate these innate immune cells with our first responders. So just like if you're in an accident, your police officers and EMTs respond to the scene first. They can evaluate the situation. They can maybe take care of the situation. If not, they then escalate it and transfer you to a hospital. And the innate immune system cells are the same way. So these cells exist to be your first line of defense. Sometimes they're enough to neutralize whatever infection is present, but sometimes they need backup. And then when that backup is activated, that's the adaptive immune system. And the adaptive immune system has this ability to develop specialized cells that can specifically target any potential bacteria or virus that you're exposed to. So these are like the doctors and nurses at the hospital who have this special ability because of their training to handle any type of medical crisis that may occur. So how does this actually work when you're exposed to a pathogen? So let's just say that you've been exposed to SARS-CoV-2, which is the virus that causes COVID-19. And what happens is that you have these innate immune cells. And some of these innate immune cells, their job is to survey the surroundings. And whenever they notice something that doesn't belong, something that's foreign, they engulf it. They essentially eat that virus or bacteria. And then what happens is they break it down, and then they have a mechanism that then allows them to present bits of that virus or bacteria on their cell surface. And what that does is that alerts the immune system, especially the adaptive immune system, that you're being invaded by a foreign pathogen, and you need your other immune cells to become active to help you clear the infection. So when that happens, you then have your adaptive immune system cells. And these are things that you might hear about in the media. They're B cells, your T cells, your antibody producing cells. Oftentimes you'll just hear people talk about antibodies, and that's because they're the easiest thing to detect clinically, but your immune system is composed of more than just your antibodies that help protect you. So what will end up happening is that your adaptive immune cells, when they kick in, will help clear the infection, and then you'll recover from whatever infection that you had. And then what ends up happening, and this is the really neat thing about the immune system, is that those B cells and T cells and antibodies will go on to form memory cells. And what those memory cells do is that they just exist in low levels in your body, but then if you're re-exposed to the pathogen, they jump up their levels, so they divide to have more of these memory B cells present so that they can attack that pathogen when you're re-exposed, and this can either prevent you from getting sick entirely, or what it can do is help prevent you from getting as sick as you did that first time that you were infected. So it's these memory cells that is what vaccines are trying to generate without you having to actually go through the process of getting the full infection. So what this ends up looking like is that if you have a pathogen, typically before the advent of these RNA vaccines that I'll talk to you about in a minute, usually what happened was that scientists took a weakened or dead particles from these pathogens, and they used them to develop vaccines. They would then inject that vaccine into the individual. This elicits an immune response that will then generate those long-term memory cells without you having to actually get the real infection. So the goal of vaccination is to get those memory cells that can protect you from the real infection without you having to go through the actual illness. Because we all know that some of these infectious diseases have very real long-term consequences or can result in death. And the purpose of a vaccination is to prevent you from having to risk being severely sick or risk dying from the infection. And vaccines do work. So this image is from the British Society for Immunology. And what it's looking at diphtheria, pertussis, and measles, which are all common. They were very common infectious diseases. Most of us probably have never really had to know anyone who's suffered from these diseases because of vaccinations. So the little syringe indicates around the time that these vaccines were introduced into society. And you can see that after the introduction of vaccines, these diseases went from being very, very common and often very deadly to being something that many of us have never even heard of. So some general important immune system points. So your innate immune system responds first, then your adaptive immune system will kick in and it has specialized cells that will help clear that infection if they're needed. And then the adaptive immune system will produce those memory cells that will help protect you if you're then re-exposed to the same pathogen. But vaccines can provide this memory cell protection without you actually having to get infected, thus protecting you from any potential consequences of the natural infection. All right, so now I'm going to switch gears and talk to you a little bit about RNA. And in science, we call this relationship between DNA, RNA, and protein the central dogma, because it's a very important relationship between all of the biological sciences. So when I explain DNA, I like to explain it as the recipe. So the DNA holds all of the instructions needed to make every single living organism. We have DNA in our cells, as do dogs, cats, plants, everything that's living. And these directions go through an intermediate called RNA. And then the RNA is used as the direct template, the direct directions to make these proteins. And each individual is composed of thousands of proteins. And what we call genes on DNA, those genes code for all of these individual proteins. So the DNA has all of this information, but it's segmented into specific areas that have the directions for specific proteins. But altogether, the proteins are what actually make the organism. The DNA is the directions. And the RNA is this intermediate that takes the genetic information from the DNA and allows it to be converted into proteins. So what this actually looks like in a cell is if you zoom in to one individual cell, inside of your nucleus this is a structure that's part of your cell. Inside of this nucleus is your DNA. And your DNA stays within that nucleus because you want to protect that original source of directions. Because if these directions get damaged, if your DNA gets damaged, that can cause cell death, or it could also cause diseases like cancer. So you want to make sure that your DNA is protected inside the nucleus where it can't be damaged. But proteins are made within the cytoplasm. So then we have this problem where you have to get the directions from the nucleus into the cytoplasm where they can be made to protein. And that's where the RNA comes in. So in your nucleus, the directions to make a specific protein are converted from the DNA into an mRNA molecule. And then that mRNA molecule leaves the nucleus and goes into the cytoplasm where it can be made into protein. And mRNA, especially in the kind that we're going to be talking about with vaccines, though the vaccine mRNA can't get back into the nucleus, so there's no potential for it to potentially hurt your DNA. So just some general important mRNA points is that mRNA is different than DNA. I think a lot of times people confuse the two, but they are different. And mRNA cannot damage your DNA. And the other thing about mRNA is that it's very fragile. So once it's used as a template to make proteins in your cytoplasm, it doesn't stick around for very long. How long is going to vary depending on the RNA, but it's overall very fragile. So it's going to be degraded and chopped up very quickly. So your body is always making new RNA to replenish and use as directions to make the new proteins when your body needs them. RNA just doesn't stick around for an indefinite period of time. All right, so then how does this all come together for a vaccine? Because obviously that's what we're all interested in. So first, I just want to briefly talk about the virus that causes COVID-19, which, as I mentioned, is called SARS-CoV-2. And viruses are unique because they don't have the ability to replicate by themselves. So without infecting a host, viruses can't make copies of themselves. So what's happening in the context of COVID-19 is that SARS-CoV-2 is infecting an individual's cells and turning that individual essentially into a virus-making factory, because that's the only way this virus can make copies of itself. And the way that the SARS-CoV-2 virus does this is that there's this little red protein on the surface, and this is called the spike protein. And the spike protein is needed for the virus to enter your cells. What happens is that spike protein binds to one of your own cell receptors and essentially hijacks your cell and then turns that cell into a virus-making factory. So that spike protein is important because I'm going to mention it a lot going forward, because the vaccines are targeted against that for that specific spike protein. So what this image is showing you now on this slide is the whole genome for the SARS-CoV-2 virus. So pretty quickly after scientists realized that there was this novel virus that was occurring in China, scientists sequenced the whole genome of the virus and were able to annotate it so that we knew what the directions were coding for. So here you can see all the different parts of the virus that are contained within these directions for the whole virus in general. But what I want you to focus on is just this region in green. And this region in green is just a small subset of all of the directions contained to make the whole virus. But it's just this region in green that makes the spike protein in particular. So to make the whole virus, you need to have all of these genome instructions. But the instructions for the spike protein are just in this little area that's in green. And that's important because what scientists have done to make this mRNA vaccine is they took just that region of the viral genome for SARS-CoV-2, just the region that makes the spike protein. And they've used that to make these mRNA vaccines. So these mRNA vaccines can't give you COVID-19 because they're missing all of the other directions needed to actually make a full virus. The only part that's present in these mRNA vaccines is this very little region here that just has the directions to make the spike protein. And as I mentioned, the mRNA in general is very, very fragile. And this is also true for the mRNA directions to make the spike protein. So to get around this issue, what the scientists did were they engineered a protective bubble, so to speak, to enclose the mRNA. And this protective bubble is called a lipid envelope. And a lipid is just another word for fat. So this mRNA is essentially enclosed in a fat bubble. And what that does is it allows the mRNA to get into your body and then actually be used without being degraded. If instead they just injected straight mRNA into you, it would be immediately degraded, nothing would happen, and the vaccines would not work at all because the mRNA would be degraded before they could do anything. So the other thing I want to mention before I start talking about exactly how these work is what's in them, because I know a lot of people have concerned about the ingredients that are within these vaccines. So both Pfizer and Moderna, which are the two currently approved for emergency use in the United States plus in other countries, is the mRNA for the spike protein. So both of them contain this mRNA sequence just for the spike protein. So again, we can't get a COVID-19 infection from these vaccines. And then all of these fancy long words are just the chemical names for lipids. Lipids are composed of a lot of different molecules of carbons and hydrogens. And these lipid fat bubbles, the formula between the two vaccines are a little bit different, but overall they both have these lipid fat bubbles. And then there are things like salt, sugars, and buffers that help to stabilize the mRNA to make sure that it can last until it gets used as a vaccine. The important thing to know is that there's no fetal cells in these vaccines, if that's an area of concern for you. There are no chemical preservatives like there are in some other vaccines, and there are no heavy metals like there are in some other vaccines, though in very, very small quantities, much smaller than could be harmful to you. But still, if that's a concern, they're not even present in this vaccine. Okay, so what happens after you get the vaccine? So you'll go to a vaccination clinic, you'll get injected in your muscle with the mRNA vaccine. When that happens, you have innate immune cells in the vicinity of that injection site. And what will happen is that those innate immune cells will recognize that that component is foreign. They're going to recognize right away that that lipid envelope doesn't belong, and they're going to engulf it. And when they do, that's essentially like the innate immune cell eating the vaccine components. So once it gets into your cells, the cell is going to all of a sudden see the mRNA for the spike protein. And the cell knows what to do when it sees mRNA. When it sees mRNA, it uses it to make protein. So that's exactly what's going to happen in these innate immune cells. The mRNA that was just introduced from the vaccine is going to be used to make the spike protein. The important thing here to remember is that the mRNA will be degraded very quickly. The estimates based on some research from the companies that produce them is that the longest they're going to be present for is 12 hours. So after 12 hours, all of the RNA is going to be degraded. And the other thing is that the RNA cannot get into your nucleus. I know there's a lot of concern that the RNA vaccines could potentially damage individuals' DNA, but that's not possible. To get into the nucleus, you have to have specific, you can think of them like access codes that will allow molecules back into the nucleus. This mRNA does not have those. And even if it did get into the nucleus, but again, it can't, but even if it did, it would then need a special protein to convert it back into DNA so it could insert itself into your DNA, and that also does not exist within our cells. So there's two really good reasons why these RNA molecules cannot actually damage our DNA. But our cells do know what to do with that RNA, and they will use it to make a spike protein. And then what will happen is that innate immune cell will present that spike protein on its cell surface, and then all of your other immune cells are going to be like, hey, this doesn't belong. We know that that spike protein is not actually part of your own body. We know it doesn't belong. And they're going to mount an immune response. Those adaptive immune cells are going to kick in, and they're going to clear whatever cells have that spike protein. So pretty quickly, the RNA is degraded, and then any residual spike protein is also going to be degraded right away. It's not going to linger. But what has happened is that your immune system has now been exposed to and trained to recognize that spike protein as foreign. And then what will happen is if you're exposed to the real COVID-19, well, the real SARS-CoV-2, your body is going to see that spike protein, know that it doesn't belong, and be able to clear the infection either before you get sick completely or before causing severe disease, which is great. And I'm going to show you now the data that Pfizer released showing how well this vaccine works. I could have also put the one in from Moderna, but it looks almost the exact same, and I didn't want to waste time talking about two almost identical graphs. But when I saw this, I was super, super excited. I think I went and showed my phone to my husband because I thought that this was some of the coolest data that I've ever seen. So what Pfizer was able to see, and what this graph is showing you, is that in red are all of the individuals who got the placebo. And the placebo is just a fake shot that doesn't have any actual protective mechanism. It's just a control. And then the people in blue are the people who got the real vaccine. And what this is looking at is how many cases of COVID-19 started to occur in these groups. And the important thing is this circled date of the 21 days, because it does take a while for your immune system to build those memory cells. So as soon as you're vaccinated, you're not protected. It takes about 21 days for your immune system to have full protection. And what you're starting to see is that at that 21 days, there's now this huge separation between the placebo group and the vaccination group, where the placebo group, those people just keep getting infected with COVID-19, and they keep getting more and more cumulative cases, where this vaccinated group is protected from disease. And from analyzing this data, both Moderna and Pfizer were able to conclude that their vaccines were about 94 to 95% effective. They also analyzed the safety data, and the data showed that these were safe. And of course, the vaccines are only 94 to 95% effective. So there are 5 to 6% of people who got the vaccine but still got disease. But the important thing is that those individuals were protected from severe disease, so they did not require hospitalization, and they did not die. So nobody in the trial who got the vaccine and then got COVID died from COVID-19, which is a huge deal and really shows how well these vaccines work. So I just want to briefly stop before I start getting into specific questions that I commonly hear about, and I want to talk about why this is so revolutionary. So the really cool thing, I was really excited because of the efficacy of these vaccines. And I don't know for sure that every mRNA vaccine would be that effective, but that data is beautiful and really is encouraging for being able to develop vaccines rapidly if needed. but also RNA is very easy to make and it's very easy to adapt. So it's very possible that mutations could occur, as we're seeing right now, that could eventually make the current vaccine iteration useless. That's not a concern currently. Right now, the vaccines that we have are protective even against these new variants, at least with the data that we have so far. But eventually, if we let the virus keep mutating, that could end up not being the case. But the nice thing about these vaccines is that it's very easy to adapt the RNA within them to address those mutations if they arise that are concerning. The other thing is that RNA cannot cause infections because it's not a live virus. Remember, it only has the spike protein, so there's no possible way it can actually give you COVID-19. So this is very safe for immunocompromised patients. So some patients who are immune compromised, they can't get attenuated live vaccines because it is a live virus. It's just been weakened to not get you as sick. That's not a concern in these cases. Also, there's some really cool and exciting work that's trying to use this same technology to target cancer cells as an alternative to chemotherapy or other treatments for cancer. And right now, some of these are in trials with mixed success, but I think that this is going to actually show that we can use this technology in humans and really open the door for being able to use it in other avenues. And then also, I think this has shown us that with resources, we can quickly develop vaccines if they're needed. And I really hope that I am never alive for another pandemic. But if it does happen, we do know that we have this technology that can be used quickly if a situation like this arises again in the future. All right, so now I'm going to start talking about some of the common questions and concerns I hear from people when they're considering to get the vaccine or not. So the first question I get is, what are the common side effects? And here is a graph from the FDA comparing the Moderna vaccine to the Pfizer vaccine. And people who did get the vaccine do report side effects fairly frequently. And these are very mild side effects, though. These are things like fatigue, headache, joint pain, vomiting, fever, or muscle pain. And the one thing I want to mention is that some people see these side effects and they think it means that the vaccine is getting them sick. But they're forgetting that what happens when you have a fever or when you have a headache or when you have fatigue from a virus is that the other thing that's happening when you are infected with a virus is that your immune system is working. Your immune system is working to mount an immune response that actually clears the infection. So the reason that you're getting these side effects in the case of these vaccines is not because the vaccines are getting you sick. It's because the side effects are occurring because your immune system is responding to the vaccine and it's producing those memory cells, and some individuals, as a consequence of their immune system responding that way, will have these side effects. But in general, these side effects last for about 24 hours to 48 hours on average. And if you need to, after you get your vaccine, you can treat these symptoms with Tylenol or Advil. The other thing I hear a lot of people expressing concern about is the fact that a lot of people are reporting that their second dose side effects are significantly worse than their first dose side effects. And before I explain why this occurs, I want to use an analogy. So let's say that you get a knock on your door one day and you open the door and you see somebody that you don't know. And you talk to this person for a while before you finally realize that they're a salesman who's trying to sell you something. Then you kick them out because you don't want to buy anything. So that's essentially what's happening with the first dose of the vaccine that you're receiving. Your immune system has never seen this spike protein before. So it's being trained to recognize that that spike protein doesn't belong. So that's what dose 1 is doing. So then let's say that salesman comes back to your door a second time. You open the door, and then that second time you're going to know he's trying to sell you something, and you're going to kick him out right away. And that's what's happening with the vaccine dose 2. We call this a boost dose. What this is doing is that you're getting re-exposed to the spike protein again. Your immune system has already seen it once. It knows that it does not belong, and it's going to react quicker and more strongly than it did with that first dose. And this boost dose is important because it's that second dose that will make sure that you produce enough memory cells that will last long enough to actually protect you from a real infection. So a lot of people are tempted to skip that second dose, but please, if you get the first dose, please go and get that second dose. These vaccines were designed to work with 2 doses, and if you only get one, there's no guarantee that it's going to work like it did in the clinical trials. And because your immune system after that second dose is responding more strongly and quickly, oftentimes people will see and report more side effects after that second dose. But again, it's not because the vaccine is getting you sick, it's because the vaccine is eliciting that immune response, which is what you want it to do. I also want to note, though, that not everybody gets side effects after a vaccine, and that does not mean that the vaccine isn't working for you. In clinical trials, there was a wide range in people who reported side effects, but all of those people who got the vaccine, whether or not they reported side effects, had an immune response that protected them. The difference is due to a lot of genetic differences in how our immune systems work, but just because you don't get side effects afterwards, don't worry that your immune system didn't work. All right, so then I also want to address the risk of anaphylaxis, because I know this is something that is a big concern for people with Moderna and Pfizer, especially since we often hear this reported in the media, and it does so in a way that's a little bit scary. So I pulled this data from a CDC advisory meeting report, and if any of you would like to see these resources directly, you can email Jerry and she can connect you with me so I can send you the links. But the CDC makes all of these things public, so you can just go onto the CDC website and download them to look at the data yourself. But these are from a advisory meeting that was on January 27th. And what they found was that out of over 9.9 million doses, 50 people reported an anaphylactic reaction after the Pfizer vaccine. And then after about 7.5 million doses of the Moderna vaccine, 21 people reported anaphylaxis. So overall, that's very, very low when you think of it. For Pfizer, it's about the risk is about 5 in 1 million. For Moderna, it's about 2.1 in a million. So that's a very, very small risk in general. And in all of these cases, 90% of them happened within 30 minutes of vaccination. And 80% for Pfizer and 86% for Moderna of the people who had those anaphylactic reactions had a history of known allergies with a known anaphylactic reaction in their previous history. So overall, I want to reassure you that while there is this risk of anaphylaxis, it is relatively low. And what the clinics are doing as a way of dealing with this potential risk is that they have every person, no matter your history, stay with it, at the clinic for 15 minutes after you get the vaccine for observation. And then if you have a history of allergies that are concerning, then they have you stay for 30 minutes after the vaccine. This way you're right on site. They can administer epinephrine if necessary. And also I want to mention that none of these people who reported anaphylaxis due to the vaccines died. They all recovered fine. So while it is a risk, it is a very low risk, especially when factored in to the risk of getting the actual COVID-19 infection. All right, so then I get a lot of questions about whether or not the vaccines will actually help reduce the number of cases. There's a lot of things in the media or people who are vaccine skeptical who are saying that the vaccine doesn't protect you from getting COVID-19. You can still pass it on, so it's worthless. And I want to say that that's not true because, first of all, nobody died after getting the vaccine, even if they did get COVID-19. So that alone makes it worthwhile. But also that even if people do still get an asymptomatic infection after getting the vaccine, and I say may because we don't know for sure yet, that data is still being collected. But even if people do become asymptomatic and can potentially spread COVID after getting the vaccine, they're going to be able to infect people for a less period of time than someone who's not vaccinated. And I want to explain why with the graphs that are illustrated on this slide. So if you look at the one on the left that has the title not vaccinated, what happens is that when an individual is exposed to COVID, the virus that causes COVID-19, what's going to happen is, like I said, their cells are going to turn into viral making machines. So slowly over time, they're going to accumulate more and more virus, and that's called viral load. Eventually, their immune system is going to understand what's going on. It's going to deal with the infection, and the viral load is going to decrease because the immune system is getting the upper hand and winning. But for that whole period of time, these individuals are contagious and could be spreading COVID-19 without realizing it. If somebody's vaccinated, they could still have a short period of time where they're infected and their body is producing virus, but because their immune system has already been trained, it's going to kick in quicker. So they're going to be producing less virus and they're going to be contagious for a shorter amount of time. So even if they could potentially be contagious, overall, they're going to have less of a chance of spreading it to others compared to somebody who is not vaccinated. So as the CDC recommends, after you're vaccinated, you should still wear a mask just in case. But no matter what, once we get enough people vaccinated, we will start to see a decrease in transmission because our immune systems are trained and we're contagious for a less amount of time. All right, so then again, another common question I see is, why do I need to get a vaccine if my chance of surviving COVID-19 is high? So this data, the US surpassed, we're almost at 500,000 deaths so far from COVID-19. And that's a lot of people who died. And even if the total risk of death to an individual is low, we know that in a whole world of people who have no immunity to this disease, that's going to be a lot of unnecessary deaths. And when you look at the data for all of the current candidates for vaccines, even if they haven't yet been approved, all of these vaccines in clinical trials, people who got COVID-19 did not die, which is a huge reason to get this vaccine in and of itself, because reducing mortality obviously is going to save a lot of people's lives. And by saying that your risk of dying is low, you're forgetting the fact that everyone's life is valuable and we can protect other people and prevent them from dying by getting these vaccines. Also, I want to mention that just because people don't die doesn't mean that they're not profoundly impacted after recovering from COVID-19. Some data shows that about 1 in 50 patients report having symptoms lasting longer than 12 weeks after a COVID-19 infection. And this is even for people who reported having mild to moderate infections. So this is not just people who were hospitalized. Also, heart, lung, and brain abnormalities, amongst other organ abnormalities, have been found in people even with mild to moderate cases. So it's important to get the circulation of this disease lower so that we can prevent having these long-term impacts. And also, even if you're young and healthy, we see that young and healthy people are dying. So you do not know what your ultimate outcome will be of COVID-19 until you get it. So you don't know if you're going to be somebody who's fine, if you're going to be somebody who has long-term health impacts, or if you're going to be somebody who could potentially die. So getting this vaccine when we know it prevents death is really important in and of itself. All right, so then the last thing I want to address before getting to people's questions is that I see also a lot of concerns with how did they make this vaccine so fast? And people are concerned that important safety steps were missed that makes this vaccine not safe. So I want to explain what they did to help get us to having this vaccine in record time. So typically what happens is when a disease is an issue and they want to make a vaccine for it, there's a lot of non-clinical research that occurs in mice and other organisms beforehand. And the thing with COVID-19 is that it's a coronavirus, and scientists have been worried about coronavirus pandemics for a very, very long time. You can do literature searches and see that scientists have been warning people for years and years and years that a coronavirus pandemic was something that could potentially happen. And this became even more real during the SARS pandemic in the early 2000s. So during that period of time, researchers were doing a lot of non-clinical work trying to understand how they could make a vaccine for SARS. And they made a lot of headway, but then the SARS epidemic ended and the funding dried up. So there was not a big push. People were still working on it, but it was working at a very slow rate because there wasn't an incentive to fund it more. So then what ended up happening is that when COVID-19 hit, and COVID-19 is about, I forget the exact number, but it's over 90% similar to the virus that caused SARS. So when COVID-19 became an issue and scientists started to realize that we were going to be in serious trouble, they were able to quickly pivot all of that early research in the SARS vaccines to adapt them to be used for specifically COVID-19. The other thing that they did is that usually when a clinical trial is being run, they have phase 1, which is some preliminary studies to see if it potentially will elicit an immune response. So they do phase 1, and then they analyze the data, and then they do phase 2, and then they analyze the data, and then they do phase 3, and then they analyze the data. And there's a lot of gaps in that process because it's happening one right after the other. So what ended up happening during this pandemic is because it was such a human health emergency, they overlapped these steps. So phase 1 still happened, but they overlapped it with phase 2. Once they had some early phase 2 data, they then overlapped it with phase 3. So instead of waiting for all of the phases to be ended, they did them in overlapping so that they could get all of the same data because they wanted to make sure the vaccines were safe and effective, but they wanted to save time. So they did not skip any safety steps. They just did it in overlapping order so that they could get it done faster. The other thing is that usually what happens is that the scientific review boards that are looking at this data do it at the end. And in this case, because of the pandemic, they have been looking at the data from the beginning. So they didn't have to have a huge analysis of going through hundreds and thousands of pages of documents at the end. They were able to do that throughout this whole period of development so that it could be quickly approved once they decided that it was safe and it was effective. The other thing is that because the government funded the development of these vaccines in production, it took away the risk from the companies of making vaccines in the factory that then wouldn't be approved. So they were able to start producing these vaccines even before they were approved to save time because the companies weren't going to be risking losing millions of dollars. The governments were guaranteeing that no matter what, we're going to pay for those doses even if we can't use them. And then the other thing is people are worried that mRNA therapies are new, and this goes along with the previous slide about concerns with safety. And I want to say that this is the first RNA vaccine that has been given emergency use approval. But as this table is showing you from a paper review that was looking at different RNA therapies, there was actually an RNA therapy used in 1998. It was approved in 1998. It was for something different. It was not a vaccine, but it was an instance where a human was given RNA for a therapeutic purpose. And in addition to this, there has also been other trials with RNA vaccines, and these trials showed that the vaccines were safe. The issue was that either the trials are still ongoing, so we don't yet have enough data, or the trials were ended not because of safety, but because it wasn't effective in preventing the disease. And obviously, we don't want a vaccine that's not going to actually protect you from getting the vaccine. So we actually have a lot of data from RNA being given to humans and none of that data suggests in any way, shape, or form that RNA is going to be harmful to an individual long term. Okay, so that was the common questions I hear from people, but I'm sure you guys have other questions. So I'm happy to take questions from you guys as well. And also, if you want some references to these topics, or you want to contact me with specific questions, my website is up on this slide. I'm happy to answer individual questions.
Liz, thank you so much. That's fantastic. What a great presentation. Very easy to follow. So thank you for putting that together for us and sharing all of your insights. We have had a lot of questions in the chat, and many of them were coming in as you were talking. So I think you may have covered some of these, but it may not be a bad thing to just sort of reinforce a few of these questions because I think we have some commonalities here. So I'm just going to kind of start at the beginning. And the first question is the J&J vaccine based on a killed virus?
Yeah, so actually that's a great question. And I thought somebody might ask me about that. So I have a slide for you. So the J&J vaccine is a little bit different. So what they're doing in this case is that instead of using RNA, they are using they made the directions in DNA instead for just the spike protein. So again, this vaccine can't give you COVID-19 because it only has the directions for the spike protein. but they inserted it into an adenovirus vector. And an adenovirus is a type of virus that causes the common cold. Usually just if you get a runny nose, it's an adenovirus. And they modified this virus so that it's unable to replicate in your cells and it doesn't get you sick. So they're using this virus just as a way to deliver the message for that spike protein. And then once inside the cell, it goes through a very similar process as to the mRNA vaccines. It's just the delivery mechanism is that attenuated, altered adenovirus that's delivering the spike protein directions.
Great. Okay, excellent. The next question is, what about the immunogenicity of the PEG, of the polyethylene glycol?
Yeah, so people who are getting anaphylactic reactions are usually doing it because of the PEG component of the vaccine. PEG is used in other pharmaceuticals, including like Miralax and laxatives. So some people do have antibodies that are against PEG, and some people then have an anaphylactic reaction. But again, a lot of us have taken Miralax for a variety of reasons and don't have an anaphylactic response. So it's not everybody who's ever been exposed to PEG previously. But those who do get anaphylactic usually are doing so because they have high levels of antibodies against PEG. So I know companies are working to come up with alternatives so that that doesn't have to be used in future iterations.
Okay, all right, great. So there are a couple of questions about side effects, particular side effects after the second dosage, and whether or not you should take ibuprofen before the vaccine or after the vaccine. I know we've seen a lot about this in the news and on social media. And then is there sort of a recommended protocol for that? So, you know, dosage wise, is it just until symptoms abate, or have you any comments on that?
Yeah, so we don't have great data. A lot of these reports are just based on theoretical information. The typical consensus, and this is true for like if you have a kid who you take them to the pediatrician, they recommend not pre-medicating. So don't take Tylenol or Motrin before you get your vaccine because that could potentially decrease your immune response. Though again, I don't know if I've actually seen great data confirming that, but just as a precautionary point, you don't need to take it beforehand, so don't. The consensus that I've seen is that if you can get away with not taking Motrin or Tylenol after because you're not super uncomfortable, then don't take it. You don't need to take it afterwards. But if you're miserable, if you have a high fever, then taking a dose of Tylenol is not going to hurt the immune response. But I haven't seen any like official CDC dosing recommendations or anything. But I do know that they recommend that if you need to, after the vaccine, go ahead and take some Tylenol.
Okay, all right, great. Let's see, we talked a bit, I think you covered a bit of this, but just to reiterate, how does efficacy work with the new strains? And I know that's a broad question, but.
Yeah, it's a great question. So you may have heard in the news as of yesterday that AstraZeneca's vaccine is currently being halted and being distributed in South Africa. And South Africa is one of the places where it has a new mutation in the spike protein of the virus. And we know that the vaccines are targeting the spike protein. The thing with the AstraZeneca vaccine is overall, it was a lot less effective than these mRNA vaccines. So then adding that mutation on top of it made it even less effective. In terms of the Moderna and Pfizer vaccine, we don't have tons of data yet. We do have some preliminary data that they've released from, they isolated antibodies from vaccinated patients and to see how effective it was. And it was still protective against the South African variant. Because even though there was a drop in protection, because these vaccines work so well, that drop did not significantly affect you. So there may be more people who potentially could get infected with these new variants, but they are still protected enough that they should get more mild disease and they will be protected from death. And we see this with Novavax and Johnson & Johnson released data a couple weeks ago from their clinical phase 3 trials. And this was preliminary data. Johnson & Johnson and Novavax both saw that the efficacy dropped for the South African variant, but they still saw 100% protection from death. So even though there's a decrease, these vaccines are still going to make sure that people survive a COVID-19 infection, which is great.
And just a point of clarification, the J&J and Novavax and AstraZeneca, they are not mRNA? Do they have mRNA? Okay.
Yeah. So the Johnson & Johnson's a DNA-based vaccine, and then the Novavax is a protein-based vaccine. So their mechanism is different. And Johnson & Johnson is not yet approved in the US. They're seeking emergency use authorization at the end of the month. And I expect that they'll get it, and I expect they'll be starting to roll out distribution in March. Novavax is still collecting more phase 3 data, so it's going to be a little bit of time before we know for sure.
Got it. So another question, in about 3 weeks, I will be deemed to have achieved immunity to the SARS-CoV-2 virus. At that time, is there an appropriate test I can take to measure my antibodies?
Not really. So there is some debate. There are antibody tests that have been used to detect antibodies after a natural COVID-19 infection, but it's unclear as to how well they'll detect antibodies after vaccination. So you could go and get a clinical antibody test that was designed for a COVID-19 infection, but if it comes back negative, I wouldn't panic because there's a lot of issues with some of these antibody tests, and they weren't designed to check antibodies after vaccination.
Got it. Okay, great. Is the difference between the required frozen storage temperature of the Pfizer and Moderna vaccines due to the different compositions of the surrounding lipid bubbles?
Yeah, that is true. Yeah, the lipid bubbles are different, and it's the lipid bubble difference that contributes to the different storage temperature.
Okay, great. Another question, could the vaccine generate a cytokine release syndrome?
So that was a concern in early phase 1 data, which is why they looked really carefully to see if that was an issue, but we have not seen that at all. And right now, I should have double checked before I started. Worldwide, last time I looked, there was only over 60 million doses of the vaccine administered. And we have not seen any of that in trials or in actual real world cases, and we would expect to see it by now.
Okay. Should folks who have allergies, particularly allergies to medicines, particularly medications like penicillin, should they be concerned about getting the vaccination?
Right now, the only allergy that the CDC says should definitely not get the vaccine are people who are allergic to PEG or who have a known allergy to sorbitol, because there is some similarities between PEG and sorbitol. Otherwise, if you have penicillin allergies or other allergies, eggs, etc., you are advised to still get the vaccine. You should consult with your allergist if you have one. They may decide that you need to be monitored for longer afterwards. But every site that is administering vaccines has people on staff who can deal with an allergic reaction if it happens.
Great. Another question, I had no side effects from my first shot. Is my immune system not responding?
Nope. So I got my first dose on Saturday because I'm volunteering to staff vaccination clinics for the next 7+ weeks. And I had my arm hurt, but that was it. But I know that my immune system is working against those spike proteins. It doesn't matter.
That's great. So will we all need to be revaccinated? And if so, can you explain a little bit about why?
Yeah, so the big question is going to be how much do we keep letting the virus mutate? So viruses mutate, it's what we know that they do. And the more people they infect, the more mutations they'll accumulate. And this is why pretty much any virologist or scientist or immunologist I know, or you know, is probably like freaking out right now because we need to get cases low. And it's not because we're worried that the vaccines we currently have aren't effective. They are. The concern is that the more you let the virus mutate, the more we could potentially eventually have a case where we don't have great efficacy with current vaccines. So it is possible that eventually the virus will acquire enough mutations that we need to be vaccinated again. And that would be because the antibodies we've already made can't recognize the spike protein anymore because it's mutated so that they don't recognize it. But the other thing is that also eventually memory can fade. The memory immune cells do stick around for a while, but how long they stick around depends on the infection or it depends on the type of vaccine. So because these vaccines are new, we currently don't know how long protection will last. Moderna came out and said that they believe their vaccine will offer protection for 2 years, but they don't know that for sure yet. That was just based on data, the preliminary data that they have. So we really won't know until we get more data.
Fascinating. One question, what can be done to boost the immune system in a way that actually works?
I'm not quite sure what that means. So in terms of like non-vaccine things, like people have variations in their immune response because of genetic differences that you can't change. But we do know that things like good sleep, good nutrition, exercise do help your immune system function the best. There is no over-the-counter compound that you can take that will boost your immune system. Anyone who says that is lying to you. If you eat a good, well-balanced diet, if you exercise, if you get good sleep, then your immune system has a good chance. And any other differences are going to be due to genetic differences that you can't change.
Got it. So I think we covered this already, but does J&J have preservatives, the vaccine?
I don't remember.
Mm-hmm.
The mRNA vaccines, Moderna and Pfizer, do not. I don't remember about Johnson & Johnson. Actually, I remember that I went and looked, but because they haven't released their documents for the emergency use trials yet, I don't know, but I should know soon. So if the person wants to email me, I can let them know as soon as the documents are released. Because once the CDC gets them, they release them to the public.
Okay, all right, terrific. And we'll share an email address for everyone to use here shortly. A couple of other questions. If a third vaccine is required for protection against a South African or another mutation, will the reaction to the third dose be incrementally greater than the reaction to the second dose was?
Maybe, but maybe not. We don't know yet. It would depend because the mutation changes the spike protein. It might not be. And again, some people are fine both times, so it's really hard to predict. But theoretically, maybe.
And do you have a sense yet, so if we do need a third dose, whether that will be just one dose, or will there have to be will there possibly be 2 additional doses?
Moderna, I know, already said that they are working on developing a booster dose specifically for the South African variant, and that would just be a single booster dose. I have not heard anything official from Pfizer.
Got it. What makes one variant more contagious and spread more quickly than another?
That is a great question. So I'm going to bring you back to this picture of the spike protein. So the spike protein can only infect cells by binding to that receptor on your cell, and that receptor on your cell is called ACE2. And some of these mutations that SARS-CoV-2 is acquiring is allowing it to bind to those receptors better. And because they bind to those receptors better, you can get infected even if you're exposed to less virus than you had been before. So let's say with the current circulating variants, you needed to be exposed to, and this is not an actual number, this is an example. Let's say you had to be exposed to 50 virus particles to get infected, to actually get disease. In the case of these new variants, because it sticks to the ACE2 receptor better, it might now be 10. And that is what we seem to be seeing with these new variants, is that they do the affinity, so the spike protein binds to our receptors on our cells better, and that allows us to be infected more easily. And then also, I mentioned viral load before, which is how much virus you produce after you're infected. Some of these new variants seem to also increase how much virus you produce once you're infected. And why that's the case is a little bit unclear.
Okay, great. If someone contracts COVID-19, is it still recommended that they be vaccinated?
Yes. Yes, definitely. So how long immunity lasts after a natural infection is unclear. But we do know there's some preliminary data from some really good labs that suggest that 10% of people are seeing that they lose protection as early as 3 months. Some people are being protected for longer, but there does seem to be a pretty steep decline eventually where you lose protection. And this is because a real virus has really sophisticated ways of suppressing your immune response. So a real virus can do things that actually impair your body's ability to make that immune response that it needs to clear the infection. And a vaccine does not have that issue. Plus, there are other things we can do to engineer a vaccine to be better than a natural infection in producing those long-term memory cells. So definitely you should get vaccinated after you've had the real infection, especially because we don't know how long protection will last for people. And also we are seeing reinfections, especially now due to this new South African variant. So it seems like the antibodies produced after the natural infection is really not cross-reactive much, a little bit, but not great with the South African variant.
Interesting. Have you seen any data that the vaccination can interfere with contraception, oral contraception?
No, I should have put that in. And I'm not sure if this person means like makes birth control inactive or could cause fertility problems. And both of those things is no, it can't. There was a myth circulating on the internet that the mRNA vaccines could cause female infertility. And the reasoning behind this was that there is a small region there's a human placental protein called cecession-1, and there's a region of that protein that has some similar amino acids to the spike protein. So somebody was saying that because of that, the antibodies that you make to the vaccine are going to cause you to have a miscarriage and not be able to carry babies. And that's not true. First of all, the overlap is very, very small. And our body, so proteins are made up of 20 different types of amino acids. And I like to think of these as like Lego blocks that are connected together. And because there's only 20 of them, every protein is going to have some overlap. Our immune system is sophisticated enough to be able to tell those apart. And also, even if this were true, this would also be true for natural infection, because in a natural infection, you also produce antibodies against the spike protein. And we know that people in the clinical trials got pregnant, and those pregnancies were maintained after getting the vaccines. And we know people who have gotten pregnant after getting the natural COVID-19 infection. So there's no scientific basis for that. It's just a myth circulating.
Okay. Lots of those out there, that's for sure. So if I had no real side effects after my first injection, maybe felt a bit shaky for about 10 minutes, should I be worried about an anaphylaxis after my second injection?
I haven't seen any statistics saying that somebody was more likely to get it after the second dose. All of the cases I've heard about have been after the first dose. So I would say the risk, if anything, is the same. But I don't know for sure because I haven't seen any data. They still recommend monitoring for 15 minutes afterwards, just in case.
Okay. And are you aware of any vaccination options for those that are allergic to PEG or sorbitol?
The Johnson & Johnson and Novavax vaccines don't have those components. So those would be great options once they're available.
Great. Excellent. All right. I'm scrolling down through all the great kudos to you, Elizabeth, for doing this. I want to make sure I don't miss any of these questions. Let's see. Does the mRNA cross the placenta and could it affect the fetus? So I think you basically already answered that one. Let's see. All right. I think we've got most of them. So oh, wait, a couple more. Hold on. Oh, that's it.
Yeah. And the thing about the placenta, because it's a little slightly different question, is that the cells that are getting exposed to the mRNA are the cells near your injection site. It's not like the mRNA is going to suddenly find its way to your uterus. That's not going to happen. So that is not a concern at all.
Great. Great. And there were several folks in the chat that asked about an email address for Elizabeth. If you want to just email development@mdibl.org, we will make sure to pass along those questions to her and connect you with her. So
yeah, and my website is here as well, and I have contact information for me on my website, which is just my name, phd.com.
Perfect. Excellent. Just a couple, I see a couple questions here. Let's just see, my chat's scrolling so fast. If I have chronic immune deficiency disease like Crohn's, am I at risk of an immune response from a vaccine?
If you're on immunosuppressants, then it's possible that you would respond less well than somebody who isn't. But there's no evidence right now. It's safe to get a vaccine if you have an autoimmune disease. I think the issue is that if you're on an immunosuppressant, then you could potentially not respond as well because of those immunosuppressants. But of course, check with your doctor before doing that.
All right, fantastic. Thank you so much, Liz. I really, really appreciate your time and such a clear presentation, very thorough. So it's great to get this information out there and give people a resource where they can get good, solid, science-based info. So thank you. And for those who are interested, we'll continue our conversation about COVID-19 in our next Science Cafe on March 8th at 5 p.m. And this will be a historian's perspective on the SARS-CoV-2 pandemic. So this is actually a former student who was here at the MDI Biological Laboratory. He's now an associate professor of history at the University of Houston, Dr. James Schaefer. And so we encourage all of you to join us for that. In the meantime, we will share the link to today's recording once it's ready for you. So we'll send that around via email. There'll also be a link on our website. And just thank you all so much for joining us this evening. I hope this was informative and helpful. And again, love to hear your feedback and any other topics that you may want us to consider for our lineup. So thanks to you all very much. It was wonderful to see everyone.
Thank you, everybody.
Thanks, Liz.